driven towards applications, and the application is context, carbon absorption and storage, and filtering out gases as they flow through, say, a factory chimney or any gas pipe or any kind of filtration that can happen and is needed. So there's a lot of chemistry involved in this talk, but I will skip through most of it primarily because I don't understand it myself, and also, but I need to still motivate you towards that. So, but before that, this work is impossible without all these wonderful collaborators. First and foremost, my professor, Prabal Kamaiti, Professor Ganapati from Chemical Engineering, IISc Bangalore, Professor Manish Kumar from Texas Austin, and our lab mates, Arun, who has finished his undergraduate with us, Nikhil, who was a postdoc, Prajesh, who was a postdoc with us before he came to IMSC, and from Ganapati's lab, Rajashekaran, who is now in Grenoble, and Wuchu Song, who was with Manish Kumar, and now he's in Berkeley. And yeah, so once again, the motivation is, yeah, this is a photo from Rourkela. So this is the Tata, like the Rourkela steel plants. And this photo was taken from one of the hills in Rourkela by our photography club. And the main thing is, whenever you need to filter out things and whenever you need to extract gases from anything, the existing methods are all solvent based. So you need to have huge chimneys, and these chimneys have to be filled with different kinds of solvents, and the gases bubble through them, and then you dissolve them, and then you extract them. But these are very expensive processes, and they require refueling and constant purchases of materials. So people thought, why not use permeable or semi-permeable membranes to get this task done? And that's the main motivation of studying polymers and other kinds of membrane-based substances towards gas filtration. So yeah, and as you can see, the scale of this chimney is probably like hundreds of feet, and yeah. So the material related works that I will talk about are already published, both of them, one which was in collaboration and one which was pure simulation and theory. So the first one is on something called a carbon molecular sieve, which was requested to the lab by Shell, and we simulated their structural properties and everything, and then we go on to study their absorption properties. And the other, okay, yeah, so this is the slide I was keeping in the poster. So we modeled everything, and I'll discuss very shortly how we did that. And the other was the collaboration with Manish Kumar, where they were actively developing membrane as we requested them to, and coming up with results as fast as they could during the COVID pandemic. And yeah, that was wonderful. So the main idea is the membranes will consist of different kinds of slits and pores of different sizes, and a gas molecule, when it passes through them, depending on the kinetic diameter of the gas, they will pass through some of them and will not pass through the others, resulting in them getting absorbed on those wedges and the smaller ones passing through. But the problem with the continuous distribution of these pore sizes is that in the bulk, everything will go through. They may get stuck at the small pores, but they will always find a bigger pore to go through. So the main motive in this fabrication techniques is to have a very fine pore size distribution consisting of ultra micropores and micropores, where these are smaller than 10 angstroms, by the way. And the ultra micropores will do the task of filtration and then channel them towards, channel out those things towards some micropores where they will be stored. So it's a very complex requirement, but nonetheless, people are doing it, and we are here. So I will skip through all the chemistry here. The only thing I will tell is that this membrane, the CMS membrane, the first one, it was synthesized some 10 years ago, and from experiments, they cleverly calculated which monomers comprised it, and they did some stoichiometry to obtain the ratio at which these two monomers, pyrrole and pyridine, are present, and that is all the information that I needed. Fortunately, they did not have any information on how they link and how they wind and how they coil. So that was a great degree of freedom for me, because but this same degree of freedom was not available in the next work because they were actively measuring everything we needed, and they even had the level of cross-linking and like the byproducts even, and from the byproducts they could again find out what exactly went in and what went out. So yeah, so in the first work we assumed that these, we first identify the sites at which they are most likely to bond, and we created various chain lengths of them and mixed them in a box, and then we did some kind of MD equilibration and got to the structure. Now this part, which I very lightly wrote of density equilibration, is the most difficult part of this process and is the cause of all kinds of like a soup of results. So I will focus a little bit on that. So the way you compress this system of particles greatly affects the final output. So in order to have a realistic picture, you need to know very well what is happening at the molecular level. And this kind of understanding was not there for this membrane for almost 10 years now. So what we did was essentially go back, constructed these the monomers. We created them in random configurations in various chain lengths and various distributions within each chain. Then we put different numbers of them in a simulation box, which is hugely sparse at the beginning, and then we apply extreme pressures and release them and do a very annealing, like unrealistic annealing process until we find a state which doesn't change anymore with time. And then we go on to verify whether this system works or not, and a similar extreme process was applied for the next, the experimental collaboration one.
How realistic are these structures in terms of experiment?
Very, like almost exact. So that's the goal of this entire work, to have a very accurate description of what is happening. So yeah. So before I discuss about the like how gases can go inside any of these pores and everything, here I want to just focus again that depending on how the pores are distributed, the density will vary, and depending on the density, every other property will change. So we need to have a good understanding of how the pores are formed. So for that, here's a schematic of a middle cross section of one of the membranes, and this is a density heat map. So you can see that there are different sizes of voids and different dimensions, and there are different ways again to compute this value. So the process we took was you roll a ball along the edge and you trace the point of loci, and then you have a surface, and then you fill that surface with Monte Carlo moves and you get the volume. But I said this in a very rapid way, but this has been very challenging and people have implemented this in various softwares. The one I used was Zio++. So essentially what we get are different pore size distributions which match very reasonably well with what was obtained in the 10-year-old experiment. And from there, we want to highlight one thing, is that they observe different kinds of peaks depending on their different experimental setups. What we have done is, instead of having different chemistry and different environmental settings and different pressures and temperatures, all we had done was taken different number of chains in the sample. So what we observe is that just by having different chain lengths, we can, in fact, get back their pore size distributions. So what that means is, whatever complexity is happening, whatever cross-linking is happening in that polymer, it essentially comes down to the distribution of these chains. And once you can identify this distribution, you can effectively reproduce the polymer. And then a similar argument was there for the experimental work. And yes, in fact, our results matched very well with their like what they obtained, and we went on to move forward. But then we went on to move to gas adsorption. But then there was a problem. I'll come back to this again. So the thing is, in an experiment, you can measure the permeability by passing gas from one side and seeing how much is collected on the other side. If you want to do that in in my simulation, it will not work because the time scales involved are huge. So to just focus on that again, so what we have to do is we have to assume that the membrane is in contact with a virtual gas reservoir, and then we have to do Monte Carlo steps to put the gas molecules in the correct locations and find how much gas is absorbed. So for that, we have to use grand canonical techniques because the chemical potential has to be balanced, and ultimately, after sufficient Monte Carlo steps, we will reach a chemical equilibrium where, depending on the pressure, the concentration inside the membrane will increase. And the final value of this concentration is what is measured in an experiment. In an experiment where concentration is measured, but that's not easy. What they measure is permeation. That's the problem. And yes, so with and like this is just highlighting that with different chain lengths again, you can get the different results. One which was with the 10-year-old experiment matches with the 90-mer and near around the 50-mer, but that's a detail which is not needed. The main point is by changing the chain length again, we get different kinds of filtration performances of these membranes. If you just look at, say, methane carbon dioxide pair in a real system, the selectivity changes, and it can be tuned to be a very good amount. For instance, in a 90-10 mixture in a reservoir, you get almost 10 times more CO2, which is what you want in a system. But then there are better polymers again. It's all about the membrane you're concerned with. And the reason for this increased CO2 absorption is explained using something called the heat of absorption. So there it's like the energy released when a CO2 comes and binds to the walls of the membrane. And here we distinctly see that CO2 consists of a very high number throughout the pressure range, signifying that it will strongly be binded to the system, while methane can flow through. So the main task for me was then to have a way to get permeabilities so that I could match with these experiments. And for that, I want to highlight the problem here. The problem is actually multiscale dynamics, because we will have different kinds of motion. For instance, the water ripples are moving in a way. There's fishes which are moving at different speeds. There is an ant which you probably can't see, but yeah, it's moving in its time. It's a very fast time scale, but the distance it covers is much lesser than the slowest moving fish here in the same video. So the same thing happens for me. For that, I have this reference where I zoom in and see a CO2 molecule, or all the CO2 molecules stuck in the membrane. And as was being highlighted earlier, the gas molecules actually interact with the walls, so the walls move. So this is a problem again where you need to have Monte Carlo moves where the walls are allowed to move, and that's a problem. And he was talking about it that day. So then what it boils down to is the CO2 molecules are essentially caged. And since they are caged, the mean square displacement is subdiffusive. So for this particular instance, the exponent is 0.71, but it can go to as low as 0.3 or whatever. It's just subdiffusive. So, and since it's subdiffusive, my problem with the theory is that I need a diffusion constant to get the permeability. And the diffusion constant can only be obtained if I have a linear MSD. So I don't have a linear MSD right now, and I do not know how to get it. So what I did was I tried to replicate the experiment. I set up my simulation box. I put a huge reservoir of gas here. I put a vacuum. I put a pressure gradient, and nothing flows through because inside the membrane it's still subdiffusive, no matter how much pressure gradient I do. So I exhaust the reservoir here, and still there is no permeation. So then this problem was not solvable in any simple way until we found that Monte Carlo can still help us if we involve timescales in every Monte Carlo step. But then if you want to include a kinetic Monte Carlo with a variable time step, you would still need to evaluate the time scale, that time step from an MD which is not available. So then we decided to keep kinetic Monte Carlo, but with a constant time. So we would assume that this is something you should not do, but we did, that you assume that each Monte Carlo step has a constant time value. And then can we do something with it? The answer is remarkably yes, we can. For that, before that, I just want to highlight that the CMS membranes interact, or any membranes will interact a lot with the gas, and you can see there's a significant amount of swelling happening. And it's not just swelling, it can also contract. If there's less gas, it can just pull in the walls of the membrane, it can contract as well. And the different gases will bind to different sites of the membrane. Yeah. So what we do is, we first do this swelling calculation. So we do this simulation. How do I play this? Yeah. So we do this swelling simulation, not swelling simulation. We just do this simulation of the gases, and then we look at the probabilities of each gas molecule staying where it is and going from one place to the other. In this snapshot, you will see that this one is completely trapped, while there are these ones which are moving around quite rapidly, but they're still in their cage, but they're still moving around, and eventually one somewhere here will be seen to cross one of these cages into a different cage. So it's a problem of the molecules hopping from one cage to the other at different time scales. So to do that, what we do is we divide this box into various subcells, and we construct this probability matrix of residence and of transition, and then we perform a biased random walk with fictitious random walkers. And the beautiful thing is, this is just a simple random walk problem in 3 dimensions without any constraint other than the bias. So in essence, it will eventually always reach a linear regime, and that is what we obtain. Now, so basically, like I said, what you should never do, but I did anyway, was I assumed that delta t, or the time step between 2 Monte Carlo steps, is a constant value equal to the step of my molecular trajectory dumping frequency, as in the trajectory, the rate at which I was saving it. So then the only parameter I have left is the size at which I can divide the simulation cell, and depending on that division, the MSD and the PIJs change, and I pick that probability matrix which faithfully reproduces my molecular dynamics simulation. Then so this is a very short 20 nanosecond molecular dynamics trajectory, and then using that short 20 nanosecond trajectory, I extend it to microseconds long, and this will take you like an hour to get. This will take you like 2 minutes to get. So it's like 30 nanoseconds, or the trajectory is actually 20 nanoseconds, and this is 2 microseconds. Like, it's a huge difference in time scales that you gain just by doing a very short MD. And I remind you that although this is Monte Carlo, the details of the probability matrix is actually all atomistic. So it includes information about charge, it includes information about LJ, it includes information about dihedrals of the membrane, and all the complex potentials that go in a fully all atomistic simulation.
About 1 minute left.
Yeah, and that's all I need, I guess. So then it's just about the results. So I do this analysis, and I get the time scales at which the system is diffusive, and it varies depending on the pressure, and it always reaches 1. No, but I don't know where it will reach 1. That's the main problem. But then once I do get it, I can now finally compute my diffusion constants, and people have in the past assumed that the diffusion constant is going to stay the same roughly in the membrane throughout for whatever pressure you give, but as it turns out, it doesn't. And yeah, so it for the experimental configuration I was comparing it with, it matches quite well. So I can go on to extrapolate it for other pressure configurations it was not explored at. And then another interesting point is, if you now go on to calculate permittivities in these systems, like you take the ratio of carbon dioxide permeability to that of methane permeability, you will get some number. Now people just do gas permeations in pure environments, because getting them in a mixed environment is very difficult. But in my simulation, I can still do mixed environments. And it turns out whatever value we can get from a pure simulation, the same setup for a mixed simulation will give a much different result. And this is a reference line which we can ignore for now. So yeah, so then there's still scope to come up with a better theory of getting mixed results, or figuring out how to do mixed experiments from both ends. And that is basically all of it. I would like to summarize that we can use existing equilibrium techniques to address non-equilibrium problems, and that very short MD trajectories at atomistic scales are good enough and strong enough to predict values at the mesoscale. And I would like to thank the funders, DST, Shell India, and all the computational resources at ISC, like the TUE, and we have our very own huge GPU clusters, so that for that as well. And any questions, comments, and critiques is welcome.
Thank you very much.
Thank you.
Well, I have one quick question.
Can I ask one? So,
Yeah.
exactly, how did you estimate when you cut off your local subdiffusion to convert it into this
Yes,
random
I don't know actually when to do it. So that's the main problem, that we don't know how long in MD we have to go to get to that regime. So that's where we keep on going and going and going until using the Monte Carlo steps. So that's fast, and we can run it for very long steps. So I no longer cut it off, but once it reaches, I can then see the equilibration there and get the diffusion. So, and that time scale actually varies quite interestingly. If you see, there's a hysteresis kind of pattern here. It's not exactly hysteresis, but it looks like the hysteresis curve. Yeah, so, yeah.
Okay, any further questions? Okay, if not, thank you very much again.
Thank you. Thank you to the organizers.